Welcome, everyone, to the February UWBC Biotech Talk. We have kind of the schedule set for the rest of the year. In March, our guest speaker will be Andrew Mealy he's a professor of medical microbiology and immunology at UW-Madison, talking about sequencing approaches to dissect viruses, host interactions during influenza virus infections. Parse will be here in April. We're not sure who the guest speaker will be yet. And then in May, our guest speaker will be Margaret Theroux, who's a postdoc researcher at the Handelsman Lab at WID, right over across the street. So it is my pleasure today to introduce Doug Gibbons, who is a senior product specialist in genotyping with Thermo Fisher. And I'm going to let him take it away.
Thank you. Yeah, thanks, Liz. Thanks, everyone. Appreciate the opportunity. I think it's wonderful what you do here. And again, thanks to Sandra. Really what I wanted to do is bring everyone up to speed from Thermo Fisher's end on what we're calling our next generation genotyping platform. And it's called Swift Array Studio. So much more than that. We'll talk about applications in the past, currently, and then in the future. Thanks, Liz. My name is Doug, and I am out of the Minneapolis area, and I've been with the Thermo Fisher Genomics Group for coming in on 6 years now. So look forward to spending some time. Rough agenda. Going to go into just a background on Thermo Fisher, some of the divisions, of course, some genetics and microarray 101, and of course, release to you the information that I came here today for, and that's the Swift Array Studio, what it does, what it's doing differently, and how it's paving the way for the future. Predictive genomics, and then, of course, we'll touch on agrogenomics and offer some time for some questions. So real briefly, who we are, we've got a number of Fisher reps, Lenny on the generalist side, and a number of other folks. I work with the genetic sciences team. Thermo Fisher is a very big company. One of the reasons I love working for Thermo Fisher is we truly are a bit of a one-stop shop. So, great example is I'll work with, the bovine community for genetic selection on arrays, and we'll still offer upstream and downstream things like extraction and third-party integration for estimated breeding values. So works out really well. If you take nothing else away from today, use me as another contact here locally for Thermo Fisher, regardless of the research that you get into. My day-to-day, my team's day-to-day, really is shown on the screen here, and that's genotyping. So we're talking high-throughput genotyping. You can see kind of the product portfolio and value propositions we bring forth. And on the far left, you see, more discovery aspect on something like an ion torrent S5 sequencer. And then as you work to the right side of the screen, for quick, easy tests, a handful of markers, handful of samples, qPCR, right? Your quant studios. So where microarray and my team reside is kind of in the middle, but a little bit closer to the 10 to, let's say, upwards of 1 million SNPs that you're looking to interrogate. And we've been around for a long, long time, legacy Appometrics. You might have heard that name. And 5 or 6 years ago, we came under the Thermo Fisher umbrella. Just kind of a background, like where might you have heard of our group or what we do? And UK Biobank is a perfect example, right? In 2013, we came up with this array, and it's so integral and not only our talk today, but population genetics all around the world. And I receive requests for this type of technology and product all the time for retrospective studies and looking at, again, the 500,000 individuals over in Europe and following that population over a number of years. So from the human and clinical side, this is a great example of our work. More broadly speaking, microarray, kind of where else do we work? And so top right, you get Repo Health for things like cytoscan. We'll talk a little bit about pharmacogenomics or PGX today. Also on the newborn carrier screening side, population diversity. And that's, one thing that you should take away from today is just the broad application of something like microarray, right? I mean, new analyzers that we're bringing out today, plus the integrated workflows, are driving turnaround time and driving down costs, even as sequencing is going down in cost. So I think that's a big point that I wanted to drive home. Back to, again, my day in and day out, it's either on the left or on the right. So something different every day for me. One day I could be calling on a clinic driving polygenic risk scores for for cardiovascular disease, and of course, providing microarrays as an enabler from a tool perspective. The next day, I could be working with genetic selection on a knockout pig somewhere in Iowa. So each one of these instances really draws forth the need and demand for microarrays. Some 101 here, genotype, most of you are familiar. Again, that's the genetic makeup. Again, when you get to the phenotype of the trait, that's really what a lot of our end users are looking to use our tools for. Along the way, we do have, again, an ability to add in markers, redesign arrays, and we have some of our top clients on the agriculture side, which will redesign on a yearly basis. SNPs, that's going to be the area of interest here. We're looking at, again, SNPs all along, and this is just a diagram of kind of the differences between synonymous SNPs and the non-synonymous SNPs. It's a nice analogy for, again, what changes the meaning of the word, or in the land of genetics, the end phenotype. Mutations, insertions, deletions. I work with a very talented design team. We're able to interrogate all of those aspects in terms of structural variation. And if it can be done, my team out of Santa Clara can certainly do it on the design aspect. Out in the lobby, there are a couple of microarrays, but the bottom line here is anything in the orange is going to be in the instrument, and then on the blue is kind of a lead up to the instrument on the bottom portion of the screen. But essentially, we have a surface covered in millions of DNA probes, and those DNA probes will eventually align to genomic DNA. The target sequences can definitely range from anywhere between 1,000 to 100,000 of variants per array. And essentially what you're doing, and part of the reason for today, is we've taken a 5-day process and turned it into 30 hours. So that sample being hybridized, all of that has been expedited, and we've shrunk down a lot of the touch points within the workflow itself. A little bit of the output, kind of take-home message from this slide is you're going to get 3 clusters in a perfect world. You've got your het in the middle, and from here you're able to essentially analyze your data with our free software, which is Axiom Analysis Suite. If you or any of your colleagues ever just want to kick the tires a bit on what we offer from a software side, that's pretty commonplace with my group to set aside a time to work with you on what analysis looks like. This is the current outfit of microarrays and formats, right? That's a big question, especially on the top right, you see ag heavy, which would be 384 format. And then the bottom left, a clinical array as a 24 format as shown. The nice part about all of these arrays, it allows for varied workflow and throughput. And remember that our customization is second to none. And most of our work these days is custom, whether that's on the human clinical side for something like pharmacogenomics, or if it's marker-assisted breeding in plants. The bottom right shows our current Gene Titan, and this is the unit that's been out for quite a number of years, I think since 2010, and we'll get to the Swift Array Studio, which we're excited to announce shortly. Workflow-wise, doesn't matter if you're in academia or industry, we're able to work with a number of workflow automation pieces, liquid handlers and such. When you're setting up your high throughput genotyping process, maybe you have some multi-drops in a closet somewhere, we can use them. Maybe you're looking to fully automate with an i7, we've got the relationship with those types of groups. Or in the case for Swift Array Studio, you want to get rid of a lot of this and centralize things into one instrument. So Swift Array Studio, a brief background. We said that the Gene Titan had been around for a number of years. The Swift Array Studio has been designed over the last 2 or 3 years. If you remember the Thermo Fisher kind of chart, we've got a lot of groups and entities, and we're proud to share that we spent almost more than any other R&D department in genetic sciences. We spent a lot of time and effort toward this Swift Array Studio. We took voice of customer, voice of engineers, voice of whoever would listen to gather and glean insight about what's worked in the past with microarrays and what needs to be improved. This is the big take home right here. We're talking from what used to be a five day or maybe at best 3 day workflow. Now we're doing it in 30 hours. And we'll get to a little bit more of why and how we did that in a moment. No sound here, just a glimpse of our promo video. And I like sharing this right off the bat because you can see, what hits home, what resonates, and it's user friendliness, right? It's touchscreen, it's automation, it's everything we kind of expected, and now you're getting it, and the field is receiving this next generation genotyping platform for high throughput, or in some cases, medium throughput or low throughput in clinical end. We've got wash stations that are fully integrated. We've got touch screens, step-by-step error recovery, as we'll see. And you might ask yourself, who purchases this sort of instrumentation? That could be a lab, somebody interested in high throughput genotyping. Could be a service provider, somebody that offers the service for labs to send out. And it could be somebody like Josh, could be somebody like Sandra, who we're in discussion with now at somewhere like this facility, right? A core. And then, of course, we have a lot of placements across clinics, hospitals, and then industry within the agriculture sector. The capacity here. So, you might ask yourself what enables, the throughput. And in this case, we've gone from running 2 plates at a time to 4 plates at a time. So there's extra ovens. There's much faster scan time, wash time, and overall a sleeker look with its own external server. I like to just share this as far as, just what it looks like to run the instrument, right? And just the walking by, what's going on. You can see what passed, what didn't. You can be sitting at home on your couch and get a text message essentially on when your run finished. So again, it's a step into 2026 and beyond. We're proud of the work that our engineers accomplished along the way out in California, where our headquarters are located. So we looked at the instruments. Here's a side-by-side comparison of Axiom 2.0, so kind of the yesteryear of Axiom arrays, and then what we're proud of here calling the Swift array assay. And the way that we were able to do a lot of this condensing is a lot of internal work, a lot of feedback from customers, and really a lot of science. And so this is essentially where we have taken that 3 to 5 day workflow and shrunk it down to 30 hours. A big selling point and value proposition we bring forth is the hands-on time reduction, right? So you take that, reduce that by about 50%. Logistically, the way things come, things are pre-mixed, shelf stable. Just the ins and outs of running a lab that just, the GeneTitan's been great, but could you? Doug, it would be really nice if you could. And so we've listened to all the if you coulds, and we've implemented and really done the work behind the scenes to make it happen. I like to share this slide because it's a nice comparison between the brand new Swift Array Studio, which is currently released for the human or clinical side across, as we'll see in a bit, pharmacogenomics or genome-wide studies. So the Swift Array Studio compared to the Illumina iScan, just some target prep differences. Throughput, right? Throughput's huge for us. And then some of the touchscreen modernizations we talked about. And we'll have a time toward the end of this talk, just so you know if you have any questions. Last couple slides here on Swift Array Studio. Again, what you're taking home today is, trusted support, the reliable, robust nature of this new instrument. Even the subtleties, right? If you did have, number one, we expect the uptime to be through the roof. But in the rare instance that you need some engineering assistance, we even have files sent to our engineer seamlessly from this instrument. So when the engineer shows up at the lab, they know what's going on, right, with the Swift Array. And the big take home, especially across high throughput customers, is the data the next day, built for speed and genome-wide performance. So very excited to share that with you. A bit of our research applications on the human end of things, predictive genomics-wise. Again, what are you going to use the Swift Array Studio for? Could be things like linkage studies, genome-wide association studies, a lot of population genetics, right? Reading the other day that we saw the population in Australia really settle 60,000 years before we thought, right? I mean, these are the type of studies that we're involving ourselves in. And of course, some candidate testing for not only drugs, but also patient stratification. I like to highlight the lay of the land of where we've been. We've talked about the red instrument on the top left. That's the Gene Titan. And now we've got the bright, shiny new toy on the right with the Swift Array Studio. We've highlighted the UK Biobank, right? What else have we done? We've taken precision medicine research array. We've added markers. We talked about our gold standard design team. This is what we do day in and day out as we work with industry and clinics and key opinion leaders to understand, right? The latest CYP2D6 markers. Like, what do we have? What are we missing? What do some of the other vendors out there have that we should be on board with? So that really leads us to, again, the Swift Array Studio on the right in 2026 and the two arrays that we have out currently. I'll answer the question you probably already have in your head is, well, okay, well, why do you only have two arrays? Answer being that we've launched the Swift Array Studio on the human end of the business first. And these two arrays were the ones that we needed the updates most critically. And so as we'll see, Pharmaco Pro follows legacy products like PharmacoScan and PharmacoFocus, right? Have no fear, though, if you are in the ag segment, we will have our ag-based Swift Array Studio assays ready around August or September of 2026. So we're waiting a few months, and then we'll have the 384 format. A little bit more about who we are, where we work, right? Credibility, right off the bat. Biobanks is a huge, huge call point for us and a big area of success, and that's just due to, again, a couple of the boxes we're checking, right? High throughput ability, content, and the ability to customize. So it matters not if you're looking at Alzheimer's or type 2 diabetes. We've probably done it in the past, and if we haven't, we can do it. This slide gets into a bit of how we do it. The best way I like to describe our custom process is quite modular. So essentially what's happening is you take a little bit of this, a little bit of that on this screen, and you customize your array. If you have custom markers that you do not want released to the public, remember, that's a majority of what we do is private custom products. As we'll see with the next slide, we do offer a good number of off-the-shelf products that are readily available for purchase. The first of which is our Pangenome Pro. This follows the likes of Pangenomics and some of the precision medicine arrays that we debuted over the last 10 years. Think of this as your whole genome coverage, your genome-wide backbone, and for a lack of a better term, a little bit of this and a little bit of that. So if you're looking to HLA type, if you're looking for a little bit of blood genomics, if you're looking for a little bit of PGX, this array offers a lot of that. I like to mention most of our arrays are imputation aware, which just means the design side of things, you're able to impute some of the markers that we may not have listed. But again, when we talk about best in class in the industry, this is one of those arrays. And as we'll get to with subsequent arrays, especially with our coverage across PGX, we're proud of what we do from an updating standpoint year in and year out. Blood genomics is another array that is an off-the-shelf product. Huge, as you can tell in the orange highlighted area, huge for blood service organizations and also for research alike. The background here is we've worked with key opinion leaders to design this array, and so blood banks are certainly open to talking, discussing this sort of technology, especially now that we have the throughput capabilities across the Swift Array Studio. Software, we've talked about software in the past. This is some specialized software beyond our typical Axiom Analysis Suite software. Kind of take-home message here is you're able to, again, obtain blood typing reports and streamline your workflows and really, ease of use, right? Integrated HLA calling. Same thing that I mentioned before can be repeated if you or any of your research colleagues have interest in further discussing this or other arrays. We can always take a deeper dive. This is one of our hidden gems. Gosh, I think the latest utilization was across canine with some gut microbiome stuff, but of course applicable across the human sector. The broad touch that we accomplish here with just the sheer number of organisms is amazing. Take-home message here is though is not quantitative, non-quantitative, and only qualitative. But just important for all of you to know that we do offer an off-the-shelf microbiome product. At this stage is going to be run on our previous Gene Titan, but in due time will be, I'm sure, accessible on the Swift Array Studio. Great. Now a deeper dive into pharmacogenomics. Pharmacogenomics, I mean, it's huge, right? We look at just testing overall, and I think the genomics workflow testing, I mean, year over year since 2019 has grown in total something like 70%. And PGX as a subspecialty, I think I read recently around 95% of patients had one or more PGX-related medication problem. And so these are the type of problems we're looking to solve with clinicians all around the world. I think I read somewhere DDI, drug-drug interactions, right? The fourth leading cause of death. Again, another reason why to get behind the impacts of pharmacogenomics. So Swift Array Studio, pharmacogenomics, it goes perfectly together. They're out in the market, and we're ready and willing to talk to anybody that has interest here. A few other stats here in terms of the global drug spend. You know, again, we spend trillions of dollars, and yet most medications are very ineffective, right? They're not effective enough. And that's what we're trying to do is find right drug, right treatment, right patient using genetics. A bit more here, just as far as who does what. It's, again, target groups when we're talking about PGX versus traditional treatments and how we compare patient genetic makeup with the drugs that they're taking. We're proud to share one assay, 3 applications. And so this is a big take-home message for folks that might be running something like OpenArray or other technologies. As far as I'm concerned, this is one of the only technologies in microarray that offers genotyping, copy number, and HLA in one assay. So use that information as you wish, but you can even see the HLA typing has the 4-digit typing and the coverage across the genotyping on some of those very important genes in ADME regions are covered. That leads us to the actual array, and so it's called PharmacoPro. So this PharmacoPro array picks up where we left off with PharmacoScan, with PharmacoFocus, and now PharmacoPro. You know, Doug, what's the difference? And the difference here is just core content, right? Core content now matched with gold standard throughput. I'm excited. My team's excited to share this with clinicians far and wide. We talked about CYP2D6. We've talked about some of the other genes important. As you'll see in a couple of slides, in addition to results in 30 hours, we actually kick out translation tables and some basic metabolizer status. These slides and recordings will, of course, be made available to all of you. It's a busy slide, take-home message. We cover a little bit of everything. Our biggest areas of success and focus, it should come as no surprise, would be areas like oncology, areas like pain management, where people are taking more than one drug, maybe more than 5 drugs at the same time. And if you go back to the why, right, it's preventing any sort of drug-drug interactions. There's challenges amongst the PGX community with dealing with pseudogenes. The idea here in this slide is we do perform some MPCR steps to free up and basically accurately target the markers that you're looking at and the calls that you're expecting. Important to say too, with any sort of new lab setup for something like pharmacogenomics, we bring forth a number of experts, which include, of course, our field teams, myself, and all of our bioinformatics groups. The aforementioned metabolizer status report. This is huge for a lot of the folks that we work with that are trying to improve patient welfare around the world. And that's a phenotype report. You can take it as it is. You can also work with third parties that make it a little bit more digestible, right? But the idea here is that you do have access to this free software, and this is part of the workflow that is included with each and every sample we run on either a GeneTitan or, in this case, Swift Array Studio. Coriell is a reliable partner that we work with, and it just kind of shows the quick look, the green, yellow, red light, if you will, on what's working, what's not working, what's to be expected, and maybe what's a bit of a surprise in terms of metabolizer status. I'm a firm believer in this, right? We're in an era where if you're taking medication, I think you should have this, right? I mean, we talk about some targets that my team works with and some reliable clients. It's children's hospitals, right? You get something like this performed for the adolescent population, and it's good for, essentially a lifetime, right? On how they're hopefully healthy now, but into the future, if they do have any sort of medication to be implemented, we can do the dosing correctly. So that's a rundown of the clinical or the human segment. A couple highlights: Swift Array Studio. It's out now. The two new arrays, the Pharmaco Pro and the update to our pangenomics array, are out now, and we offer our own lab at Thermo Fisher to run any sort of pilots, and in due time, I hope to work with a number of the clinicians around the university here to bring this to reality on the clinical side. Agrogenomics, I know this is a big ag hub. I come from an ag hub. I'm a Michigan State Spartan, so I don't know if I can say that too loud, getting close to March Madness, Lenny, but yeah. Agrogenomics. Okay, wait. Why am I seeing a red box? What happened to Swift Array? Remember, Swift Array Studio, August, September timeframe of this year. So currently we're relying on our GeneTitan, a robust instrument, just not the 30-hour time frame, more like 3 days, 5 days. But we have service providers that can, again, provide off-the-shelf arrays, custom arrays, anything that can help drive your marker-assisted selection program. Same thing, ditto from what we talked about. Where are we focusing? 10,000 SNPs, up to 1 million. As you'll see in a bit, we have a lot of custom work where folks will start high, something like a 96 format array, and then they'll whittle that down into something like a 384 format array, which we haven't heard in the past in the human segment, right? 384, ag. So high throughput, and it's important to know that we do have that ability to redesign anytime. Multi-species we'll talk about in a bit, but that's another hidden gem, kind of like the microbiome array. In a situation that you, your colleague, or maybe your PI has interest in genotyping a specific species, and maybe you don't have the throughput. Maybe you don't have the grants that you need. We're able to combine multiple species on one array and hopefully make that a little bit more economical for you and your lab. Good question is, who uses our technology? Short answer, a bit of everyone, right? Industry is very, very big for us, whether it's bovine breeding groups, whether it's the poultry folks, whether it's helping scientists reintroduce coral reef that are more or less susceptible, I should say, to global warming and some of the warming water temperatures. I mean, these are all types of projects that we work with. I think the area that we're most excited to get into is certainly a little bit more into the plant space and a little bit more into the aquaculture space. Historically, we've worked quite well with the livestock world and expect to continue that partnership along the way. I mean, big numbers here, right? The one that sticks out to me is the 94 something billion, right? It's just the sheer number of SNPs through this ICBF. So kind of the Cliff's Notes version of ICBF. It's the Irish Breeding Association, or the Cattle Breeding Foundation of Ireland, and they've set out a monumental goal to genotype every head or every cow in Ireland. And so this is a huge partnership for us. As we'll see in subsequent slides, where they find a lot of value is stuff we've talked about, right? The one thermo or one-stop shop approach. So being able to work with us for collection tubes all the way through extraction, and then the genotyping calls for genetic selection. They've also enjoyed, like others, the ability to basically design an array, and what you design is what you get. So you don't have any sort of SNPs or variants missing from your particular array. I've heard it said before, and I've seen it in person, the fidelity of our arrays. So you could design an array 10 years ago and run it, today, it's going to perform the same. So we've got clients that will, hey, where have you been? I haven't heard from you in 5 years. Doug, new grant, I want to run another, 10,000 samples with you on that array I designed a few years ago. No problem. Takes around 4 weeks for a rebuild. Everything from our microarray standpoint is manufactured in Singapore. Our office is in California in the Bay Area, and then they can be shipped to one of our warehouses. Custom flexibility, we chat about it in the past. Our design team can take anywhere, probably around 8 to 12 weeks as a ballpark figure to design a new array. And then you would just want to add on that 4 week for build time or manufacturing. So it bears repeating, most of our work is custom. We do it better than anybody. And we've talked about multi-species, and now you kind of have the full picture of the flexible, affordable, and scalable aspect. Microarray, even from the top of the talk, you get it. It's a high throughput tool. It's one that you want to look at a lot of markers and then whittle that down into kind of more of an everyday prediction model for your species of interest. That's what we do. And that's a bit of what you see here with our 96 format array. I mentioned for the audience here, there's a couple arrays to take a peek at out in the hallway. And folks will use that tool from the 96 format as a screen, vet polymorphisms, figure out which probe sets work the best. Maybe you don't need 6 replicates, maybe you only need 2 replicates, and then eventually you pare down to maybe, I don't know, maybe 50,000 markers. And if you're a geneticist at a large bovine company, breeding company, you would use this tool to, again, genotype your head at a certain cadence. We do offer a mini 96 format. And so all that does is if you have a 384 format and you still don't have enough samples to fill a 384 well plate, no problem. We also can make that mini 96 format to serve your needs from a throughput standpoint as you grow. So to summarize here, routine use, breeding decisions, day in and day out. What you design for is what you get on your array. And again, the ability to run the same array over a number of years and still get the same results. Whereas a good 80% or so of our work is custom, we do have devoted a large catalog of off-the-shelf or what we call expert arrays. And we call those expert arrays because we work with all the experts to design them. So the last couple of years, we're highlighted by a lot of movement across direct-to-consumer testing, for instance. What type of poodle do I have? Is my poodle going to be plagued with some sort of disease, right? So these are the type of things that we've designed with our Canine HD array. Parentage testing, disease testing, things of that nature. And we've recently, as we'll see in a bit here, announced the release of our feline array. But in addition to the direct-to-consumer market for companion animals, which has been great for us, we work day in and day out with the wheat folks, whether that's big government institutions, whether that's academic groups, or of course, more industrial. We've got an array for a majority of the commodities, of crops, of fruits. At the end of this talk, you'll see my email. So if there's any really relevant questions as far as what we have, what we don't have, Doug, I'm happy to answer those. The aforementioned feline genotyping array, proud to announce we work with Dr. Leslie Lyons out of University of Missouri to develop this array. It is in a 96 format. And again, the reason being because we have, as you can see, around 650,000 polymorphic markers. It includes multiple breeds, as you can see, and the the collaboration with Leslie was awesome, and she's one of these researchers and leaders that we have so much respect for, and really has notoriety all around the world. So my colleagues and I have hit the ground running with this array, working with anyone from animal shelters to academic institutions that want to maybe set up shop as maybe kind of a direct to consumer market. If that's a small vet clinic, we offer this same sort of service and technology to that small animal vet clinic. This should be a bit of a reminder of kind of how we build our arrays. You take kind of a modular approach. Illumina had a 63K array that was very important for a lot of our end users on the feline side, that those markers were included in our array, and they are. We also worked with OMIA, and certainly a number of Bengal populations that Leslie Lyons knows much more about than our group, but we were happy to work with her there. And then, of course, the ancestry traits and disorders that we talked about in a similar array on the canine side. Now we have it for public release on the feline side. Yeah, a couple more slides here. Here's repeating that we have our own microarray service lab out of River Falls, Wisconsin. So you do have the opportunity to send us samples for anything we talked about today. We use that option as a nice pilot for researchers that have interest in everything we talked about so far. Again, I've been working with Josh, with Sandra, as far as the future here at University of Wisconsin. Clearly, the ag component of Swift Array is not out quite yet, but as we get closer to the third quarter of this year, I'm sure we'll have further discussions. And one day soon, I hope to see something like a Swift Array studio floating around these halls. Microarray, our service lab, you are able to send either tissue or extracted gDNA. And then once we receive the gDNA, we're able to turn around results for any sort of pilots in anywhere between 5 and 7 weeks. So you'll receive the genotyping reports, and we'll have a nice wrap-up call with some of our experts if there are any questions on copy number, library files, or any sort of output that you're expecting. I see on the third column there's something known as Axle, and this is our tool that we're able to actually convert files to an Illumina format. So if you're used to something like an Illumina format on their arrays, we're able to mimic that for you in your output. That kind of wraps up what I wanted to chat about today. Clearly, we'll have some time for some questions, but hopefully you can tell from my excitement that this is, yes, it's been a long time coming with Swift Array Studio. I'm happy to share the new technology with you, especially with the workflow enhancements, like 50% reduction in hands-on time, and truthfully, just the throughput. So as we get closer towards the summer, we'll have an update on the ag side. I've got my contact here, which I'll leave up if you do have questions from anything in the past that anyone has run on Affymetrix legacy products, anything current that you have interest in, and of course, as we pave the way for the future, any new lab setups or aspirations for genotyping at a broader scale. Good. Any questions here in the audience? And I think Liz will probably channel any questions my way in the chat. Okay, sounds good. Good. Well, I appreciate everyone's time. I look forward to staying in touch. And if there's one thing I can leave you with, it's Thermo Fisher. It kind of goes back to what we talked about. There's no one thing that we have that might solve all of your problems, and we really pride ourselves in that one Thermo total approach. So if you have something like a need for lower number of samples, lower density, we've got qPCR, we've got OpenArray, and maybe that particular technology and box opens some doors as you grow your throughput in business to one day get into the Swift Array Studio, or even like a gateway box like the current Gene Titan out there. So thank you again to Liz, to Sandra, and to the whole university for having me. Grab some food out there. Yeah, thanks, guys.